Hello students, as this is my first session, let's begin by solving the previous year questions which will be highly helpful in all biology related exams. Let's start. The first question is to match the following. In list 1, the scientist names are given and in second list their discoveries are given. We all know nucleus is discovered by Robert Brown, so we can match that. The ribosome is discovered by George Palmer. The chromatin is discovered by Fleming, and the disc-shaped sacs or cisternae near cell nucleus. What is the disc-shaped sacs? That is the Golgi body. So Camillo Golgi can match with the disc-shaped sacs. That is Golgi bodies. Uh, in his names, the Golgi bodies is named based on the scientist name Camillo Golgi. And the ribosomes, we know that ribosome is different in prokaryote and eukaryote. In prokaryote, the ribosome is 17S and in eukaryote, the ribosome is 18S. And the ribosome has a large and a small subunit. In case of prokaryote, the large subunit is 15S and the small subunit is 13S. This 15S subunit is made up of rRNA and protein. It consists of 23S and 5S rRNA and 34 protein. 13S small subunit consists of 16S rRNA and 21 protein. In case of eukaryote, it consists of 16S. This is 16S subunit and 14S subunit. The 16S is made up of 28S, 5.8S, 5S, rRNA is 28, 5.8, 5S, and plus 45 proteins. The small 40S subunit consists of 18S rRNA and 30 proteins. The function of ribosome is protein synthesis. It's helpful in protein synthesis. And there is more chance to get confused between the prokaryotic and eukaryotic ribosome. So try to remember this. Let's move to next slide. Second question is, mesosome in a cell is a membrane-bound vesicular structure. No. Chain of many ribosomes attached to a single mRNA. What is this? Chain of many ribosomes attached to a single mRNA is polyribosome. That is, this is mRNA and ribosome is attached to this. It's known as polyribosomes. So it's not the answer. Special structure formed by extension of plasma membrane. Extension of plasma membrane. This is known as mesosome. So it is the correct answer. So mesosome is the extension of plasma membrane. Don't forget that. And next. In next slide, it's also a match the following. Students, the match the following questions are very easy to catch the marks. So don't skip this kind of questions. The first option, first column, histone, nucleosome, euchromatin, heterochromatin. We know that the histone, it's positively charged protein. Histone is positively charged protein which is attached to negatively charged DNA. So we can match this. And DNA nucleosomes, DNA wrapped around histone octamer. The nucleosome is formed by DNA wrapped around histone octamer. This is the, consider this as histone and the DNA is wrapped around the histone. And there is another histone and DNA is wrapped around this, like this. And this is the linker DNA. This is the linker DNA, and this structure is known as nucleosome. And next is euchromatin. Euchromatin is densely packed chromatin. So we can match like this. And heterochromatin is loosely packed chromatin. So we can select this from option D is correct. B for C. Hmm. C is one. Oh, sorry, there is a correction. The euchromatin is loosely packed chromatin. Euchromatin is loosely packed chromatin. Please note that. Because it is a actively euchromatin is a actively expressing region, so it's available all the time. And heterochromatin is the non-expressing region. It is highly condensed region, so seen at nuclear periphery. Heterochromatin is seen at nuclear periphery, highly condensed, so it is a densely packed chromatin. Please note that. Option A is adhering junctions facilitate the cell to communicate with each other. It's not the correct answer. Uh, what is adhering junction? It's keep two cell to adhere. That is, if this is the cell and it's the neighboring cell, this adhering junction helps to adhere between the cell, or it performs cementing to keep the neighboring cell together. Next, tight junction. Tight junction helps to stop substance from leaking across a tissue. Is it correct? Yes. Tight junction helps to stop substance from leaking across a tissue. That is correct. Then tight junction helps to perform cementing to keep neighboring cell together. No, it's not correct. Then gap junction helps to create gap between the cell and tissue. Now this is wrong. What is the function of gap junction? It helps to facilitate or communicate the neighboring cell. That is for transfer of ions, small molecules, etc. So gap junction helps to communicate between the cells. Next slide is match the following. The list one F1 particle histone axon cistern. Case of we can blindly match that axonym is related to cilia, and histone particle is related to chromosome, and the cistern is related to Golgi apparatus, and F1 particle is related to mitochondria. What is F1 particle? In mitochondria inner membrane there is apparatus F0 F1 or oxysomes, which helps to generate ATP in electron transport system. It helps to create ATP. Okay. Okay, that's about the today's session. Please let me know if there is any correction or suggestions is needed, so that same can be taken forward in the upcoming section. Thank you.